My name is Barbara Qurollo, and I am an assistant research professor here at NC State in the docs department. I primarily work in the vector-borne disease diagnostic lab, where I help with diagnosing vector-borne diseases primarily in dogs and cats, but we do some wildlife samples as well. And then my projects stem out from that, which usually involve improving the diagnostics for detecting these mostly blood-borne pathogens, and also stem from identifying new organisms and then the epidemiology around them and the diseases they cause in animals. I recently was awarded an AKC grant, and this will be for a project that's going to study spotted fever group rickettsia organisms in dogs. And we're going to focus on a new organism that we identified in 7 dogs over the course of several years in our lab. Spotted fever group rickettsia are tick-borne pathogens, and probably the most famous one that most people are familiar with is the one that causes Rocky Mountain spotted fever. So that's Rickettsia rickettsii. And this is a pathogen that can infect people. So people get this disease as well as dogs. And it can be really serious. Animals and people can die from it if they're not treated in time. And so there are other spotted fever group rickettsia in the United States in particular that can cause disease in people, but as far as we know, not really as severe as Rickettsia rickettsii. And then these other spotted fever group rickettsia can infect dogs as well. But from what we know, that they don't really cause a lot of clinical signs that we can detect. And so what's unique about this project is that we identified this new spotted fever group rickettsia, which we haven't named. And these dogs were really sick, kind of like if they had Rocky Mountain spotted fever, but it definitely was not Rickettsia rickettsii. And so all of these dogs got similar clinical signs. And in fact, 2 of them unfortunately died. They weren't treated in time. The other dogs were treated with doxycycline, which is the drug that treats most of these rickettsia organisms, and they did fine. So we are really interested to know how prevalent this organism is, what ticks transmit it, where else it can be found. And so this project is about, first of all, trying to develop a better diagnostic test to detect different spotted fever group rickettsia in blood. Spotted fever group rickettsia infect endothelial cells. These are the cells that line the blood vessels. And so when we detect this organism in blood, we're really detecting it in any endothelial cells that are shed into the blood. So it can be really difficult to detect this organism, trying to get high enough loads to detect. So we want to be able to develop better tests to look at all these other spotted fever group rickettsia in dogs to see if they are also getting infected with not only this novel one, but other ones like people we see in people. And then we would like to ultimately culture this new organism, and that's going to require us getting fresh samples. So we really need to identify this again in the next couple of years and hopefully be able to contact the veterinarian and get a large enough blood sample to try to culture from a clinical sample. And so we have different avenues with some shelters we're collaborating with to try to identify dogs early on that have the clinical signs, detect the organism, and have enough blood to culture it. And if we culture it, then we're going to sequence the entire genome, and we should get a lot of information about the organism from that. We're working with Dr. Melissa Nolan at University of South Carolina, and then Ricardo Maggi here will also be helping to develop the improved diagnostic test. So, and then another aspect will be testing ticks. So we have collaborators in Oklahoma and South Carolina, and we will collect ticks here in North Carolina and try to identify the organism.